Right, good afternoon again, everyone. So welcome to the second part of the very short lecture series on biophysics. So um, in the first part, I've basically set the stage. I've introduced you to the context of biology, focusing mostly on molecular biophysics. And now in the second lecture, I'm going to show you a couple of selected theoretical and experimental examples, just to give you an idea of where the physics fits in and how we can get really quantitative, very interesting information about these systems. Right, so basically the only theoretical approach that I'm going to use, or the method, is in the context of quantum biology. So to show you where quantum mechanics can describe biological systems. This is quite a fascinating field. And then the experimental methods are different methods that can be used to manipulate single biomolecules, and methods to control or to assure biomolecule nanoparticle interactions. We'll also say a few words about superresolution imaging, which has revolutionized imaging of biological systems, and then a few words about single molecule spectroscopy. Right, let's look at quantum biology. This is a very young field, in fact. Um, uh, but it depends on how one looks at it to determine how young it is, because Erwin Schrödinger and Niels Bohr could be considered two of the fathers of this field, and that's already many decades ago. But the real activity in this field, the real advances have been made in the past 20 years mainly. So what is quantum biology? That is the field that studies and investigates processes in living organisms that cannot be accurately described by the classical laws of physics. So in other words, you try to describe a physical system, biological system from a classical point of view, and then you realize, well, there are some features that you cannot describe, and you have to invoke quantum biology in order to describe that. There's a recommendable headline review that was published some 3 years ago, and I was one of the leading authors of that paper that describes or gives a perspective of how can we envision the future of quantum biology, and with a focus also on some technologies that can be inspired by the field of quantum biology. Let me give you a few examples. So here's a list of systems that have been um, argued are the context for quantum biology. Most of them, well, basically all of them have been theoretically described, and from a theoretical point of view, quantum effects have been proposed to be significant. Um, and for some of them, there have been some experimental efforts, but experiments in this field are really challenging. That's pretty difficult. Um, okay, so let me say the most profound evidence of quantum biology is in the context of photosynthesis. So there we have real evidence that quantum biology takes place. And what I'm talking about here is not quantum mechanics at the molecular scale, because otherwise everything in the universe is quantum, but I'm talking about the next level of organization. So can non-trivial quantum effects be used by these systems to promote their biological function? So, in other words, does quantum um, mechanics play a physiological role? And in photosynthesis, this is definitely the case. So, photosynthetic systems, they consist of an array of proteins that harvest the energy from the sun. So they will absorb photons, and then they will transport this photoexcitations to a reaction center. And in all these processes are um, evidence of quantum effects, specifically quantum coherence, and throughout the whole photosynthesis process, you have coupled electron and proton tunneling. The same with respiration. Respiration works in a pretty similar manner as the so-called reaction center in photosynthetic systems. And here in respiration, you have four different macromolecular machines, which are responsible for coupled electron and proton tunneling through the membrane to the opposite sides of the membrane. Olfaction, that is a phenomenon of smelling. So how does it come about that we can smell so many different odorants, so many different smells? And it has been proposed that quantum mechanics plays a role in this in order to give the very large diversity. And the mechanism that has been proposed, it has to be tested experimentally, but it does make sense from a theoretical point of view. You have a donor molecule, an acceptor molecule, you have electron tunneling from the donor to the acceptor, and this is coupled with some specific vibrations in the system. So some vibrational states or phonons will couple to the electron, and this will facilitate electron tunneling, which will then lead to the sense of smelling or olfaction. Magnetoreception is another very interesting field which describes how many birds, migratory birds, and also many other organisms have a sense of direction. For example, birds that migrate from the North Pole or Northern Hemisphere to the Southern Hemisphere in one season, and then late in the year will migrate back, so thousands of kilometers that they will fly, and they have an extremely good sense of direction. So they make use of the magnetic field of the Earth, and the mechanism that has been proposed is based on the fact that these birds only fly when there is light present, so during the day, or if you do experiments in a lab when there is light present, or they only have a sense of orientation when there's light present. This mechanism must involve a photoactivated molecule. So what has been proposed is that in the eye of the bird, there's a molecule called cryptochrome, and when that is activated, a delocalized state will form between the cryptochrome, which is a donor, and an acceptor, and it will transfer an electron, but not completely. So delocalized radical state will form, excited radical state over both of these two molecules. And due to coupling with the Earth's magnetic field, there will be a hyperfine interaction, so switching between a singlet state and a triplet state, and this hyperfine interaction is modulated by the magnetic field of the Earth. So theoretically, very nice mechanism, experimentally extremely difficult to test. DNA repair and enzyme catalysis, there's also proton tunneling involved with those mechanisms. Anesthesia, yeah, also a very difficult field because, yeah, you have to put patients to sleep and you should not kill them during the process. So to investigate that is very difficult, but some distinct quantum phenomena, or let me say non-trivial quantum effects specifically, have been proposed there. And then even cognition, so our sense of consciousness, and even our sensing, so how action potentials are fired through the nerve cells, which is also connected, obviously, to cognition or consciousness. Yeah, it has also been proposed that quantum effects play a major role there. So there's a lot of speculation in that field. Also, obviously, a very difficult field of research from an experimental point of view, but very, very interesting. Okay, so these are a few examples. In many of these systems, let me say basically all the systems except the DNA repair, respiration, photosynthesis have not been experimentally tested. So in these three systems, there is experimental evidence, and the best evidence is in photosynthesis. So that's also why this is the most vibrant field of research in this domain of quantum biology. Okay, quantum biology has very interesting long-term applications in diverse fields such as sensing, health, the environment, information technologies, and here are just a few examples of that. And these are key enabling quantum technologies that have been part of the EU Horizon 2020 program during the past 8 years, and massive amounts of funding by the European Union was given to the best research worlds in the research groups in the world that have tried to realize these quantum biologies. And I'm quite an avid supporter of bio-inspiration or biomimetics, because if you just look at these enabling quantum technologies, there are superb examples in nature that can do this, which humankind cannot yet do. If you consider the first one, single photon, so to design a single photon detection system that operates at low voltage. We cannot do this yet. We are getting closer, but we cannot do it yet. But the eye does it every time you see something, or any time it senses something. Photoreceptors in general can do it in nature. Light harvesting complexes of photosynthetic systems can do this. All of these systems are single photon sensitive, working at a very low voltage or very low potential or gradient. Okay, and then nitrogen fixation at room temperature and pressure. We can't do this yet, but there are certain types of bacteria that can do this. So let's go and learn from the bacteria how they do that, and that might inspire us to design our own technologies for our own purposes. The chemically powered transport of ions and electrons across nanometers with low dissipation. Well, this is done in every cell and most compartments or subcellular, the organelles, subcellular membranes, where ions are transported across these membranes with negligible, basically no dissipation of the energy. All of this is chemically powered. How to produce hydrogen from protons without the use of noble metals and at 0 volts standard hydrogen electrode. Well, we can't do this yet. We really want to do this to promote the so-called hydrogen economy and to counter the global warming. But many organisms do this. They do it just naturally. They just ingrained within their DNA to do this. Okay, and then the last example here is to separate charges with near unity quantum efficiency. So we would like to do this, using photons. So using photons to generate charges, separate them, and have no energy loss. Photosynthetic systems do this, but we strive really hard to get this done in photovoltaics. So yeah, just a few examples of how much we can learn from these natural systems to advance our own technologies. Okay, this slide, or a modified version of this, I've shown in the first lecture. And here I would like to focus on this very fuzzy border between the classical and the quantum regimes. And what is fascinating is the fact that proteins or macromolecules are on this border. So you can describe molecular systems like proteins from a classical point of view, from a quantum point of view. You can only get a full picture when you include both of these approaches. And we need tools from both sides in order to understand the function, the biological function of these systems. From a bottom-up approach or a top-down approach, using quantum approaches to promote the functions, or the other way around. But this relationship between the classical and the quantum worlds, this is from a physics point of view an extremely interesting question. And especially to investigate this in biological systems, because as I described in the first lecture, these are dynamical systems. They have all sorts of so-called disorder, so intrinsic disorder, and then especially changes in the function that are imposed by the environment. So this border between the classical and the quantum worlds is very ill-defined, and it's time dependent. It can change, for example, into this or to that, depending on the environment. And this is also very interesting to investigate, how to tune the quantumness of a system. Okay, let's zoom into light harvesting proteins. So from the leaf here, you can see the cell. So this is a light image, a micrograph. You can see the green grains there, which are the chloroplasts. And if you zoom into one of these chloroplasts, you can see membranes, which are called the thylakoid membranes. They are regions that are stacked and some others that have a lower concentration. And inside these membranes, that's where basically all the photosynthesis takes place. Photosynthesis itself is a remarkable process. If you think of the fact that these systems use light to produce food, and they get their energy from light, and they extract the protons and electrons from water. Oxygen is given off as a byproduct, and it uses just cheap materials in an extremely efficient way to produce its own food. On the top of the food chain, virtually all other organisms are dependent on these systems that can make their own food. Okay, and then at the heart of this process are these light harvesting systems. So this is a protein which is densely packed with pigments. So pigments are the chromophores, which I have listed in this slide. So this system consists of 3 identical subunits. Each subunit is a protein. So this is a ribbon, which was introduced in the first lecture, this gray part. So you can see the 3 alpha helices that span the full membrane. And this protein binds, one protein here binds 18 pigments or chromophores. So there are 2 types of chromophores, the chlorophylls and the carotenoids. So 14 chlorophylls, 4 carotenoids in a space of about 4 by 4 nanometers. This is really extremely dense. And the consequence of this high concentration is that quantum states are formed. So these quantum states are called molecular excitons, which give rise to coherently delocalized excitations. And here is a simple explanation of this. So let's consider two-level systems. So you can consider 2 of these chlorophylls that are close together so they can couple with each other. And let's say they are identical. So they have identical energies here. We have the ground state energy there. We have the first excited state there. And they couple with a coupling strength of V12. Okay, now if this coupling strength is much greater than the coupling between the excited state or the electronic transition dipole moments with the environment, with the environment, I mean the phonons, the vibrations. So if this electron-phonon coupling is much smaller than the coupling between the two-level systems, what will happen is that your wave function of this coupled system will be delocalized over both of these molecules. So this is the wave function of molecule 1, wave function of molecule 2. You can see here molecule 1 is excited, molecule 2 is in a ground state, where the opposite is happening in this case. Molecule 1 is in a ground state, molecule 2 is in excited state. So this notation means that this molecular wave function is delocalized over both of these. Okay, so this is one thing that happens. You have the delocalization, but you also have splitting of the energy. So you now have 2 energy levels which are split by twice the interaction energy. Okay, and this has huge consequences. It's very beneficial to the organism that this happens. And here are 4 of the main physiological benefits. So the first is that due to this splitting, the absorption cross-section is extended because now you have more energy levels that can be populated. So this gives rise to more efficient light absorption. It also gives rise to faster energy funneling, or due to the delocalization, which I will explain in the next few slides. It also gives rise to faster energy transfer and to less trapping and loss of the energy. And these last 2 can be compared with trying to play soccer with a golf ball. I don't know if any of you have tried to do that, but you can just imagine what will happen. You are not going to get very far. The golf ball is going to get stuck every couple of meters. So why is that the case? It is because of the size of the gold ball compared to the roughness of the landscape. So very similarly, in these systems, you have the so-called energetic landscape, which is a rough landscape. And you have the size of the excitation when, if it's confined to a single molecule, it is relatively small and it can be trapped inside this energy landscape very easily. So when it's trapped, chances are great that it will be lost. And that is what happens in all solar cells that we try to make. The excitation is relatively small and you have all these defects in these systems. So you can try to get rid of all these defects by heating up the system. And this is quite cost intensive. But these light harvesting complexes, they function at room temperature. And they have a lot of defects, or what you could call the biological equivalents of the defects in these semiconductor or even the organic solar cells. So the way in which these systems are now designed is not to try to get rid of all the defects. That is an extremely expensive approach. No, they make use of these molecular excitons. Just increase the size of your excitations. It's like now playing soccer with a soccer ball. So the soccer ball, the size is now bigger than the roughness of your landscape. So the soccer ball, you can kick very far into the field. And the same now with these molecular excitons. They are large enough that they can't get easily trapped within this energy landscape, and they can easily travel along the landscape, or they can travel along the gradient. So here's an example of, or a conceptual example of such an energy gradient. So you have the energy on this axis, and you have all the different pigments that participate in the transport of the energy. So if you consider the energy localized at each site, at each molecule, then you would need 7 steps from the inlet to the outlet. But now if you make use of these excitons, say that these 3 are coupled, these 3 are coupled, these 3 are coupled. Okay, and now you start at the inlet, and instead of 7 steps, you now need only 4 steps to go to the end. So the energy funneling or the relaxation along the gradient is much faster due to the delocalization of these excited states. Okay, so to give you another idea, okay, if you deal with quantum mechanics, you want to solve the Hamiltonian, because once you have the Hamiltonian, you basically have a complete description of your process. And solving the Hamiltonian for these biological systems is not a trivial task. A lot of approximations have to be made, but it can be done using an open quantum systems approach, where the system of interest, say the excitations, that is your system here, and it interacts with the environment, which is the phonon bath, that is all the vibrations in the system. Fast vibrations, slow vibrations, all of that. So you have the interaction between the system and the environment. And the full Hamiltonian will then be simply, well, let's say simply, you divide it up between the system and the environment, and then the coupling between the two. Okay, and then we can expand each of these systems. So the electronic Hamiltonian, okay, if you consider the matrix notation, so on the diagonals, you will have this term. This term will describe the off-diagonal components, where you have the interaction between different pigments or different transition dipole moments. And this is the coupling term between the transition dipole moments. Okay, and you also have the Hamiltonian describing the electron-phonon coupling, so the coupling between your system and the environment. And for that, you need this coupling term between the nuclear modes and the excitons. Okay, so you can build up your full system like this. Okay, and then you can model experimental spectra. For example, here is a fluorescence spectrum, the emission spectrum, where you have the steady state population at each site, so each molecule that contributes to this fluorescence, and the fluorescence line shape, which you can divide up into all these terms. For example, you have the dipole strength of each molecule involved. You have the exciton energies, the Stokes shift, which describes the relaxation or the reorganization energy due to the coupling of the electronic states with the environment. You have a line shape function, you have the decay. So all these terms depend on your electronic Hamiltonian, and you can fit the spectra, do simulations, and eventually you can get an Hamiltonian that is satisfactory. But it's not trivial, because it's a very complex system. For example, here you have 42 interacting chlorophylls, you have non-equivalent site energies, you have a very complicated spectral density. The spectral density describes the interaction between your system and the environment. And you have static disorder, which just means that the site energies and the coupling between the molecules are time dependent. Okay, but it has, the Hamiltonian has been calculated for a lot of these light harvesting systems. And from this, you can get a good model for the cascade of the energy within such a system. So what you see here, okay, this is now the LHC2, the main light harvesting complex of plants. Here are all the central atoms of all the chlorophyll molecules, and they are all labeled from 601 to 614. You have the chlorophyll A's and the chlorophyll B's. Okay, so here are all the sites. So you can say that this is the spatial axis, this is the energy axis. And you can now, from this model, predict what will happen upon excitation of one of these molecules, how the energy will be delocalized and how it will be flowing throughout the whole system. What are all the different time scales? So in other words, you can get basically a complete picture of the interaction and the flow of the energy and the whole cascade that will happen in this system. Yeah, so this is one very small part of the big domain of quantum biology, the type of information that one can get in these systems. Okay, if one considers different realizations of the disorder, so every time you run your simulation, you will get a different result, which does not mean that your simulation is wrong. This is just a sign of the dynamic nature of these systems. So these are dynamic systems. Every time you excite one of the chlorophylls, the energy may end up at a different site. And these are all the different spectral forms that one can measure, or actually that have been measured, and different spectral forms that can be simulated, and one can then get a very good idea of how the energy is distributed across the whole pool of chlorophylls that are participating. Right, this is a little bit about quantum biology. Let's now get to a few experimental examples. Okay, and I'm basically just going to flash a few examples, and I hope that you'll be able to see how powerful these different techniques are, the broad range of applications that one can use them for. Okay, so in the first set of selected experimental methods, I'm going to show a few methods which I find pretty cool, which you can use to manipulate single biomolecules. The first one is called DNA origami, and they are basically molecular breadboards. So you can use them to make or to use DNA as a building material to build basically any type of form or any type of structure. So here are many different types of structures that are all made from single strand of DNA. You can also make the breadboard in such a mesh form, and then you can put some molecules on top of them. And because of the charged nature of the DNA, you can then selectively put some molecules at specific sites. So in order to get exact orientations and distances between these different molecules. Here's another example of using very short strands of DNA at designated positions, so called some DNA staples. Okay, and then they used a complete genome of a virus so complete DNA genome, and they by using these staples or DNA staples, they could make a smiley face of this genome of the virus. So you can just see that you can use your creativity and do anything if you want to play around with it, but the practical value of this is really immense, because now you can you can basically glue the biomolecules you're interested in at very specific places and then start to investigate the interaction between them. Okay, with optical tweezers, you can do pretty similar things to some extent. You can also manipulate biomolecules by immobilizing them using a focused beam of light. And the Nobel Prize in Physics was given 3 years ago, or it was shared half between this application and then some femtosecond laser spectroscopy application. Okay, so you might all know how optical tweezers work. It's just a balance of forces that will give rise to immobilization of a microparticle or a nanoscale particle. So these days they can go down to lower than 10 nanometers of the dimensions of a particle that they can immobilize. And here are a few examples of what you can do with that. So here are two beads that are trapped in two different optical traps, optical tweezers, and these beads bind a strand of DNA on this side, a strand of DNA on that side, and a protein in between them. So now you can very finely control the position of these beads using the optical tweezers. So you can then pull these two beads apart, and by doing that, you are pulling the protein apart. So this is one way of investigating protein folding, or the forces that proteins can withstand, or you can, yeah, all these mechanical properties of the proteins can be investigated in this way. Another application is that you have DNA bound to this part, to this bead, and you have a protein bound to a strand of DNA to the other part, and then you are pulling the DNA through the protein and investigating the interaction between the DNA and the protein. Here's another example of how a ligand, so that's a type of protein that interacts with another molecule. You can investigate how these interactions work. Another application this method has been used very much is for motor proteins, how a protein like walks on, on a cytoskeleton, and you can then pull it and measure all the different properties, the physical properties of these systems. Okay, so this was optical tweezers. Another very recent method, which I find quite cool, is that you can use light to weigh a protein, to measure the weight of the protein. And how this works is that you use scattering, a light that is scattered off the protein or off a nanoscaled object. And it's not just a pure scattering, but the scattered light is then interfered with a back reflected light. This forms an interference pattern, which is then a very sensitive measure of different properties of the system. You can use it to weigh the molecule. You can use it to track a single molecule within a living cell, for example, so single particle tracking. Okay, another method is by using metallical nanostructures by localizing or enhancing the radiation. So then you can use this, for example, to confine the light to nanoscaled regions, and you can look at the interaction between the metallic nanostructure and the biomolecules, many diverse applications there. Here's an example of a nano antenna that was coupled to a near field probe of a microscope. Okay, so the light will then be localized by this nano antenna very close to the surface, forming a hot spot, and then protein was brought into this hot spot, and you can manipulate many of these properties in remarkable ways. So here are a few applications listed of using metallic nanostructures. Specifically, they are creating surface plasmons, localized surface plasmons, which have a very broad range of applications. Okay, you can also use some other biomolecules. This example I've shown you in the first part of the talk, but now imagine using an optical tweezer or DNA origami to, to guide the exact positions of these molecules with respect to each other. Now you can start to investigate interactions between them as a function of distance. In a very controlled environment, you can add chemicals, you can change the environment in whatever you want. Yeah, you can bring light in, you can bring electric field, magnetic fields in. You can combine a whole range of different methods to investigate these biological functions, the interactions between different biomolecules in a very deep quantitative manner. Okay, a second type of method, which I've already alluded to, is by using metallic nanoparticles, which would then create surface plasmon resonances. And in fact, the field of interfacing biomolecules with nanoparticles is a very active field of research. Also from a physics point of view, interfaces between different types of materials is also very interesting. And now if you bring biomolecules in, that makes it extra interesting, I would say. Okay, so surface plasmon resonances are formed, or localized surface plasmon resonances are formed when the dimensions, or a specific dimension of your metallic nanoparticle, a metal like gold or silver, when this dimension is smaller than the wavelength of the light. So what is then going to happen is that the electrons, the free electrons in the conduction band of this metal, are going to oscillate with the light as it's transmitted through this metal. So the oscillation of this electron cloud will be in resonance with the electric fields of the light that will be transmitted through the material. And this then gives rise to a localized surface plasmon resonance. So if you have a gold nanorod here, for example, okay, it will, and you shine light on it, or it absorb light, then it will create these so-called hot spots at the tips of the nanorods. And these hotspots can they concentrate or localize the electromagnetic field, or the light in this case, to concentrations that are orders of magnitude greater than just simply focusing the light to the diffraction limit. Okay, now if you would put a biomolecule in there, for example, yes, again, the example of LHC2, you can drastically change the photophysical properties. For example, if this biomolecule is far, there will be relatively no change in the emission intensity, but as you bring it closer, you can significantly enhance the emission intensity. For this specific system, up to 2 orders of magnitude. For other types of systems, 3, maybe even 4 orders of magnitude, you can change the emission intensity. When you bring it closer again, there will be electron transfer from this system into your metal, and you get very strong quenching. Okay, yeah, what is fascinating about these gold nanorods is as you change the length, but you keep the width the same, you change the aspect ratio, you can shift the spectrum, the absorption and the emission spectrum of this system quite drastically. So you can literally tune the color of this gold nanorod through all the colors of the rainbow. Okay, so this nanorod has 2 axes along which the electrons resonate. So these are called the longitudinal axis and the transverse axis. And the longitudinal axis is the one that can be tuned in this case. So you can tune it in such a way that the aspect ratio will give rise to absorption properties and emission properties of this nanorod that correspond to that of your biomolecule of interest. So in this case, we have the LHC2 spectrum, the absorption of the nanorods, the blue one that coincides with the absorption and the fluorescence of LHC2 in order to optimize, to maximize the resonance. Okay, and what we have done was to spin coat a layer of these nanorods, and then on top of that spin coat some of these biomolecules, and then investigate the interaction between the two. We managed to drastically tune both the radiative and the non-radiative rates. So you can see here the radiative rate, that is the emission rate, which is here just for normal LHC2. Here it is increased to 2 orders of magnitude greater when you couple this LHC2 or bring it into the hotspot of a gold nanorod. Okay, so on this axis you see the enhancement factor in the intensity. On this factor you see the lifetime of the fluorescence or the emission. And as the radiative rate increases, the non-radiative rate is also significantly changed. So you drastically tune these photophysical properties, and in this way you get to understand some of these intrinsic properties of your molecular systems. This is just one example. There are so many different examples of interfacing these biomolecules with nanoparticles. Okay, the third example, I'm going to introduce you very briefly to an imaging technique that has revolutionized imaging, especially in a biological context. That is super resolution imaging on nanoscopy. So that is using light to get beyond the diffraction limit. Okay, so what is the diffraction limit? Let me just refresh your memories. So if you focus light, or any electromagnetic radiation for that matter, but let's focus on light now. So you can try to focus it as tightly as you can. You will always have to face the principle of Huygens, which says that every wave front can be divided into some source points, and every source point is every point on this wavefront is the source of another wavelet. And now if all these wavelets overlap in a far field, they create a new wavefront. Okay, and this wavefront is what determines the diffraction limit, because because of the diffraction between all these different wavelets, a diffraction pattern is formed in a far field if you focus your light. Okay, and the diffraction limit is roughly half the wavelength of your electromagnetic radiation. So if you stick with a visible light, at the edge you have blue light. That's the shortest wavelength of about 400 nanometers. So you can get a resolution of at best 200 nanometers using light if you don't use tricks to go beyond the diffraction limit. So now there have been people who were awarded the Nobel Prize some 7 years ago who developed some methods and some tricks to use light to go beyond the diffraction limit. And these methods can be divided into 2 categories. One is called, or is based on the principle of stochastic localization microscopy. I'm going to very briefly introduce you to that. PALM or STORM are the abbreviations used for the main method. And the other method is called deterministic depletion or STED microscopy. Also a very nice technique, but I'm not going to go into details now. And what is fascinating is that both these techniques can get an order of magnitude improvement in the resolution. So they can go down to 10 nanometers of resolution if it's being done right. So using light, just using light. Okay, so how the first set of techniques work is, say that you have a point source. A molecule can be considered as a point source because it's much smaller than a wavelength of the light. So you raster scan across such a point source, then you get this raster scanned image. And when you do a line scan across this image, you get something like this. So here are all the counts of the different pigments, and you can then fit this histogram with a Gaussian, and this Gaussian that the peak position will have some variance. So this is the uncertainty. Okay, so if you do this only once, you have a pretty big uncertainty. But now if you do this several times, you can decrease the variance. And because the variance scales with the inverse of the square root of the number of times that you repeat this. So this is a way to localize the molecule. So when you scan across this image, you can localize the molecule because you know that chances are great that it's in the center of this histogram, but obviously there's some variance. But now if you repeat this several times, you decrease the variance, and you can increase the certainty of knowing that your molecule is in the center of this image. Okay, so this is the one principle that you can drastically increase the localization by just repeating the measurement several times. But this is not the only thing. Now, because localization is not the same as resolution. So here's a wild field image. So you want to get the resolution of all the different parts inside. But now if you use a fluorescent marker and you label all the different parts inside, and now you want to image this, everything, all these, let me say the diffraction patterns of all these different emitters is going to overlap, and you will just get one big blur. So that's not going to increase resolution. So you can do this as many times as you want. This is not going to work. So this one principle of localization alone is not going to give you super resolution. The second principle is that you have to work with sparse subsets of these emitters. And then you have to find a way of switching them on and off at random times so that you can, so that they are far enough apart in order to be resolved. Okay, so if you can do this with a lot of different subsets, you can build up your whole structure because you can then resolve all these different emitters, which will then appear at different times. So if you do this enough times, you will be able to resolve this whole structure much beyond the diffraction limit. Okay, so this is a basic principle of Storm and Palm. Yeah, here's just one example that using a normal wide field fluorescence method, which is limited by the diffraction limit, you get just this one big blur. But with Storm, you now see that there are actually two emitters there. And here's another example of wide field and super resolution spectroscopy. Okay, so that can bring it down to about 10 nanometers resolution. The other technique, STED microscopy, can similarly, using different tricks, also get you down to 10 nanometers resolution. So now, since the Nobel Prize was awarded, so within about 4 years, Stefan Hell started to, well, actually immediately started to combine the two methods. So yeah, actually just to go back here. So this method is used, is based on the emitted light, the fluorescence light, while this method is based on tuning the excitation light. So it means that you have two different approaches, and Stefan Hell has now combined these two by exploiting the changes you can make on the emitting side and at the excitation side. And by combining that, he developed a new technique called min flux that can now go down to a resolution of 1 nanometers using light. This is really astounding. So this was also listed as one of the top 10 breakthroughs in physics in 2017. And here's an example of using the palm and the storm. So the traditional super resolution techniques that in this case have not resolved the structure entirely, but in this case using min flux, you can go down to the nanometer spatial scale. So this is really remarkable. Okay, a super resolution imaging is actually based on the on the technique of single molecule spectroscopy. Single molecule spectroscopy is a much broader field. This is the field that I am specialized in, and I see that we don't have that much time yet, so I'm just going to say a couple of things about this, and then I can end this lecture and open the floor for questions. The single molecule spectroscopy, this is the technique where you want to do spectroscopy on single molecules, and mostly biological molecules. Because of the dynamics of these systems, a lot of this dynamics is averaged out in an ensemble. But now if you can investigate these molecules one at a time, you can get access to the full dynamic range of these systems. So you can extract a lot more information about them. But this is not too trivial to do, because you have to imagine that you want to get spectral information of a single macromolecule at the diffraction limit, which gives you a volume of roughly 1 femtoliter. And within this femtoliter, this is obviously not the only molecule. There are about 10 to the 20 other molecules within this volume. So you have to be able to differentiate between the information of the single molecule and all the other background information. Well, a quite simple way to do this is to, go outside of the cell, isolate the protein or DNA or whatever you're interested in, and mimic the environment and dilute this environment, the aqueous environment, enough so that when you focus your beam of light on this molecule, you will have only one molecule within this focal volume with a very high probability. Okay, and since this molecule, well, you can then decide that you can label this molecule with a fluorescent label, so that only this molecule within your focal volume of 1 femtoliter will emit radiation, and then you can use this emitted radiation to investigate all the dynamics that take place. Or you can investigate autofluorescent molecules, and there are not many of them, but that is what I am specializing on. All these photosynthetic systems, they are autofluorescent because these proteins bind photoactive molecules that are responsible for absorbing photons from the sun. And this photoexcitation should then be converted into useful forms of energy. So these systems are intrinsically fluorescent, which makes them much easier. It means that all the changes you see in the fluorescence are not due to the fluorescent labels that you have added to your molecule of interest, but all these fluorescence dynamics relate to intrinsic dynamics that take place. If you want to read up more about single molecule spectroscopy, I have written a few review papers already about that. And I think let me stop here. I actually wanted to show one example of that. But yeah, we can maybe show that at the end if there's still time and interest left. Okay, so thank you very much for staying tuned in until the end of these 2 long lectures. I would be glad to receive any questions.
Thank you very much. Thank you very much. And so any question? So we would like definitively to have your voice and to see what you think about all those breakthroughs, because that's so exciting. I mean, it's really a field that I didn't know much. Last year, so I get to know a bit more. And there is definitively a lot of potential as well in Africa, I see. So for all our students, so please ask your question now. You have a chance, or in the chat as well. You can do so.
You have a chance now, but if you want to give it a bit more thought, you're most welcome to send me an email. On the front page of both of these lectures on the slides is my email address, so please don't hesitate to do that. But yeah, for now we can take this one question.
Please, Stefan, go ahead. Yes.
Please go ahead, Stephen.
Hello.
Yeah, we can hear you, Stefan. So then what's your question?
Thank you for the nice lectures. My question is just one. I would like you to explain how does the carbon emission from motor engines affect the the absorption of photons by the chlorophyll in plants? Thank you.
Okay, okay. Now, this is a very good question. Actually, if you just consider the overall process of photosynthesis, they do that they manage to do two things simultaneously that are part of the global issues that humankind try to face. So they sequester carbon dioxide. They actually use carbon dioxide from the atmosphere. They use that as a building material. Yeah, so yeah, that they use it as a building materials. The trees that you see outside, they are made mostly from carbon dioxide. That is the way in which nature stores carbon dioxide. And how does it do it? How do photosynthetic organisms do this? Well, they use light from the sun as a source of energy, and then they use water, well, most of the organisms, to extract the protons and the electrons that will eventually drive processes such as storing the energy from the sun in the form of ATP. So they are using extremely cheap materials in extremely efficient ways. Yeah, just consider light, water, and carbon dioxide. How much cheaper can you get in making these extraordinary products in the form of biomass eventually? But if we consider solar energy, we can just use the same principles to design our own systems in very efficient ways, or actually focus much more on um, using cheap materials and at the same time take CO2 from the atmosphere and use that as in whatever way you want to use that. Okay. Does that answer your question, Stephen?
Is it okay, Stephen? So I think it was like a good answer, very complete. Then
Mine was about let me talk of carbon monoxide.
Yes. So what's your question about that?
Is that as you can get to see the smoke that is being produced by engine in Africa? Yeah, because most of us usually use motor vehicles, and that smoke is really polluting the environment. So
Right.
my question was about that carbon monoxide. How does it affect the chlorophyll in terms of how it absorbs the photons from the sun?
Okay. Yeah. So I don't know much about carbon monoxide. Yeah, I'm not a chemist for that matter. But as far as I know, carbon monoxide is not used in the process of photosynthesis. I can be wrong, but I don't think that plays a role.
But it is not used there. But how does it affect?
How does it affect photosynthesis? I don't know. I don't know processes at that level. I just know one thing which we have to appreciate is that nature is really a marvelously dynamic system. So in whatever way mankind distorts nature, or well, if there's, for example, a volcano eruption, which is also a huge distortion in nature, nature always has a way to find a balance again. It might not be the balance that it was before the perturbation, because yeah, we see all these effects of climate change. So nature tries to reverse the effects that mankind does due to climate change, the pollution and all of that. But it's very dynamic, but it's still limited. So that's why there's, in my opinion, there's a big fuss about climate change, because this is something real. This is something which nature can't completely counteract. Yes, there's a question.
yeah, Abacus, if you don't mind. Is it okay to read it?
Yeah, is that one about the sound waves? How are sound waves or photons or odors or flavors or touches detected by a sensory organ and converted into electrical pulses that provide the brain with information about the external world? Well, welcome to the world of biophysics. These are exactly the type of questions that we ask and want to find the answers for. So yes, many of these processes, especially if it concerns the odors and the flavors, we don't know yet exactly all the details of that. There are some mechanisms of how that works. I'm not too much into that area. I can say more about the sound waves and the photons. So how, you mean for, yeah, convert it into electrical pulses so the information can go to the brain. Okay, let me give you the example of the eye, for example. Okay, so your eye can detect single photons. There's a whole cascade of events that will then turn the photoexcitations into pulses. And the main protein that's involved in converting the pulses from the eye to pulses in a nerve cell are called G proteins. The G proteins are a big class of proteins, really fascinating proteins, and they are actually involved with translating many different types of impulses that the body or the cells, or actually the proteins in the cells get, and translate that into pulses that can eventually be read by the brain. But these are very complex processes. It's normally a whole cascade of events, a lot of proteins and other molecules that are involved there. I would say Google this question and see what information you can find, or even on YouTube, you'll be able to get really fascinating information. But yeah, as I said at the beginning, welcome to the world of biophysics. This is how you have to look at biological systems. It's like, how does physics fit in? And for most of these systems, it fits in at the molecular level, because that's where most of the fascinating physics takes place. Okay, next question. How long have you studied biology after your master's in physics? Well, in fact, I never studied biology. I studied the biological context as I felt it was needed in the field. So when you do research in biophysics, as in all other scientific disciplines, you focus on a very narrow field. So my field is photosynthetic light harvesting. So obviously I had to get acquainted with the biological context of that field. And in order to understand the function of proteins and DNA, I had to read a bit more broadly. But I mean, I will not be able to give a first-year biochemistry class. I don't know enough biology to do that. And even less, I won't be able to give a biology class because of my lack of training. I can approach these systems from a physics point of view. And that's also why I would like to encourage you, if you have a physics background, don't be afraid to dive into the field of biophysics, because you are not going to become a biologist. You're going to think like a physicist if you have trained like a physicist, and you just have to know enough biology to understand the context. And yeah, well, as you discover more about the context, you get more excited to learn more about it. That's how you broaden your horizons and your scope.
Exactly.
No, there's no other question. Oh, there's another
The last one.
I'm interested in knowing the interactions you consider when investigating the DNA binding proteins with optical tweezers. Okay, so one example of that is what I showed in the first lecture. You have this enzyme called, yeah, see if I don't have the name in front of me, I forget it. Okay, it's part of the ribosome. It's this enzyme which zips open the part of the DNA so that the messenger RNA can then attach to that antisense part of the DNA in order to copy that information from that part of the DNA. So this protein that zips open the DNA, that is a very good example of interaction between a DNA and a protein. So, and yeah, you can use optical tweezers to investigate that. You can then control the distance between the DNA and the protein, and basically see in real time how the DNA is zipped open and then closed on the other side again. Now, these are just fascinating molecular machines.
Yes, very good.
Yes.
It answers
Oh, yeah.
all the question. Very good. And then maybe can you also elaborate a bit more when you say with the molecule, you look at the spectroscopy with single molecules, so it's macromolecule, or how do you isolate, how do you prepare your observation or your sample? How do you work on that?
Okay, so do you mean how do we isolate the molecule or the protein?
From the protein, or is it maybe by calculation? Is it theoretical? Because you said that you were looking at this aspect. So
Yes. Okay, so in order to isolate proteins, well, the crystallographers, the protein crystallographers do it every day. So there are very standard procedures to do that. And that it's a task of biochemists to find the recipe that works best. And I actually have a molecular biologist in my group who has isolated some of these, but many of these recipes or the isolation protocols are pretty simple. If you have had basic chemistry training, so first year practical chemistry, you should be able to do it if you can follow instructions, which is always a good thing. So then you can isolate your proteins. And yeah, then to mimic the environment, these are also very standard techniques to use, and the biochemists again give us the guidance for that. So you can, if it's something new, you can read up in the literature. If some biochemists before you would have isolated proteins, so then you just use the same chemicals as they, and those are the chemicals that will then mimic the natural environment. So from that point of view,
all the way to the molecule as well.
Yeah, so we call it single molecule spectroscopy. Maybe a better term would be single macromolecule spectroscopy, because the protein is a macromolecule. It's considered a whole unit. But obviously you can then extract some parts of this protein. Say this protein binds some lipids or some chlorophylls in this case, then you can look at the individual chlorophylls as well, if you want.
Okay, very good. And maybe as well for the CRIPS technique, or are there any kind of development that you follow up as well? Because this is very useful as well in your field, I guess, to understand with the CRIPS, to look at the DNA level and have possibility to recombine and work on those type of technique.
Sorry, I did not completely get your question.
The possibility to understand how to cut one part of the DNA, I mean some part of the message, to be able to reconstruct as well. So are you working on this development?
No, I'm not working on that myself. It's a quite big field of research, but I would say most of that part is very well established. Just because it is, one could say, it's at the essence of life. It's understanding how the code of life works, the central dogma of molecular biology. But I'm not involved with that research myself. But yeah, I know some people who are actually actively looking at DNA using some of these false manipulation techniques, including also optical tweezers. There are many others, for example, AFM. You can also use bind your DNA to the AFM tip, the cantilever, and bind the other part to your substrate, and then you can pull your DNA or protein apart. This is a very similar way as using these two beads, or binding your DNA to two beads and pulling them apart using optical tweezers. There are many of these manipulation techniques, and I know people who are doing that, who are actually still involved with some type of research. The state of the art these days is not to use these techniques in isolation, but in combination. For example, using optical tweezers together with super-resolution imaging, combining some of these techniques. And the development of the techniques and integration of different techniques, that's one very active part of biophysics. So if you would go to a biophysics conference, you will see that there are normally several tracks of people working on the development of new methods, because many of these methods are specific to the investigation of biological molecules. And I also find this a fascinating field, because then you can use your skills as an experimental physicist to develop new techniques and demonstrate that on certain biological molecules and then discover new interesting information about these systems.
To understand how they work, the structural biology. So that would be rather in that field. Very good, excellent. So I think it was a very, very exciting and fascinating presentation and session that we have. So we thank you again. So definitively some follow-up as well in that field. And then I'm just checking if we should all gather back. So in this main room, so Ketavi is or should be about to close this breakout room, and we will so first stop the recording.